Thank you all for. So, like Torsten said, my name is Jeroen de Ridder, and I'm an assistant professor in the Delft Bioinformatics Lab at the Delft University of Technology. And I'll be presenting this paper that came out in February this year. So it shouldn't come as a surprise to this audience that the genome is not linear, right? The genome is a 3D structure. And one of the reasons is that we need to fit 2 meters of DNA into the compartment of this tiny little cell nucleus. But it has also all other important implications, for instance, for gene regulation and other functions that are carried out by this genome. For instance, the 3D genome facilitates long-range gene regulation by engaging in these promoter-enhancer loops. This is an image of one of the famous examples of the beta-globin locus. This is 4C data with a viewpoint at this HBB1 gene. And what you can observe from this figure is that there's actually long-range contacts, about 30 kb, with these hypersensitive sites in the LCR. So this is data from Wouter de Laat's lab. It's also been shown that functionally related genes actually co-localize for co-transcription. And this is sometimes referred to as these transcription factories, so specific sites in the cell nucleus where transcription occurs. And this is a picture of that. But at the very least, this shows that there is communication between regulatory sites and that this communication may occur across very large distances and even sometimes between different chromosomes. This 3D conformation is not exactly stable. Especially at the local scale, there's actually a lot of dynamics and variation going on. However, at the global scale, it turns out that this genome architecture is actually quite stable. And this has, for instance, been shown by looking at these topologically associated domains by the Bin Rand's lab, and looking at the boundaries of these stats. And they show that, for instance, between 2 different cell types in mouse, these boundaries overlap in human and also between mouse and human. So these boundaries are actually very stable. Similar results have been obtained also by the Van Steensel lab, where they looked at Laminar-associated domains, or these LADs, and they showed that actually most of the genome consists of these constitutive LADs, which are present for at least 4 different cell types. The 3D genome also plays an important role in diseases. It plays a role in, for instance, determining the translocation partners in cancer genomes and shaping the copy number aberration landscape in tumors. So the question that we asked is, can we study the effect of these long-range chromatin interactions on the co-localization of cancer-causing viral integrations, in this case the mouse genome? And we want to do that to better understand the repertoire of target genes that are really targeted by these integrations. So why do we study these integrations? Well, they are frequently used in a technique called insertional mutagenesis. This is a forward genetic screening technique to figure out potential new cancer genes. So it works as follows. So we infect mice with, in our case, the murine leukemia virus. This virus integrates itself into the infected cells, in the DNA of these cells, and as a result, a tumor develops, and we can take out the tumor, sequence it, and determine the location of all these integrations. Now, when these integrations occur in the vicinity of a gene, they can hyperactivate this gene, and the reason is that these genes contain, they have very strong enhancer and promoter sequences in the LTRs that are located on both ends of the provirus. Now, not all of these integrations will be driving the tumor formation. Many will be background mutations or passenger mutations. And we need to distinguish between those, and that's why we typically repeat this experiment many times and then aggregate the results, and then we look at these clusters of integrations, which are called common insertion sites. And they represent the driving insertions, and there is apparently selective pressure on having insertions in this locus. Yeah, and that's how we can distinguish them from these background insertions. And now we can look in the vicinity of these common insertion sites to determine the target genes of these integrations. Now, there's one important assumption that we have made when analyzing these types of data, and that is that when we do this clustering, that we assume a linear genome. Yeah, but we know now that the genome is not linear, and that's why we actually asked, are there hotspots that consist of linearly dispersed insertions that are actually in 3D spatial proximity through the chromatin conformation? And if this is the case, can it be the case that insertions target genes that are actually not next door? So those are the kinds of questions that we asked. And to answer that, we looked at a very large insertional mutagenesis screen that we published a couple of years ago. It's, I think today it's still the largest one around. And it's based on the leukemia virus, which induces leukemia and also lymphoma. We have about 20,000 insertions in 933 tumors. And we combined those data with Hi-C data that we obtained from Bin Rand's lab, actually the previously published paper by Dixon et al. And it measured Hi-C contacts in ES cells and in cortex cells, and we only used the intrachromosomal data in this case. So just to refresh your memories in terms of the Hi-C, what does Hi-C measure? It measures DNA-DNA interactions by cross-linking the DNA, fragmenting, ligating, purifying, and then sequencing, and then mapping those to the genome, and then you get this contact matrix, where for any locus, two loci in the genome, you can measure how frequently they were in contact with each other. Now, what you can see from this matrix is that there's a very strong diagonal band. The same is actually observed in this figure. This is the distance, this is the Hi-C contact. And this band is actually an indication that two loci that are very close by in a linear genome also have much higher Hi-C contact, irrespective of the 3D conformation of the genome. So if we want to study the 3D conformation, we need to actually get rid of this, what we call linear distance bias. And to do that, we devised a normalization strategy, which is a rank-based normalization strategy because it doesn't use the typical assumption of normally distributed Hi-C values. So how does it work? Well, for any entry in this matrix, we actually replace it by its rank relative to the superdiagonal on which it occurs. Yeah, and these superdiagonals, they represent the Hi-C scores for bins that are a certain distance d apart from each other. Yeah, and by replacing it with its rank relative to this superdiagonal, we actually take into account the distribution of Hi-C scores of two bins that are a distance d apart from each other. Now, if we do that and we move the superdiagonal to the top right, then these superdiagonals get shorter and shorter, and we need to actually correct for that. So that's what we use this zigzag pattern to take that into account and then pull a couple of these superdiagonals. And the reason that we can do that is that actually as the distance increases, this linear distance bias reduces. So that's why this is appropriate. Now the result that we get is something like this. This is for chromosome 2. And what we observe is this really strong plate pattern, which points to the two-compartment model of chromatin, which actually has been observed also by other authors. So it seems that our normalization method picks up the right things. Now we want to use these data to study the relation between insertions. And to do that, we segmented the genome into 200 kb bins and distinguished between three types of bins. The bins without any insertions, bins with only a few insertions, so those represent bins that probably harbored a couple of background mutations, so there's insufficient evidence for selection here. And then the recurrently inserted bins, which have a lot of insertions, and there's a lot of evidence for a selective pressure on having an insertion in such a location. Those are similar to the common insertion sites. And so we have three bins, and then we can look at all the combinations of these three bins, and then look at the Hi-C contacts between them, and then you get this plot. So these are the combinations, this is the Hi-C value, and what we observe is actually that for the cases where both bins are recurrently inserted, there are elevated Hi-C scores for those. Yeah, so they're more co-localized than the others. We can also do this for the remaining chromosomes. Yeah, that's what I just said. We can do that for the remaining chromosomes, that's indicated by the colors, and then look at all the combinations of these boxes in the previous box plots. That's given on the x-axis here, and the y-axis is the difference in median Hi-C score. And now the observation to make from this figure is again that the most significant elevation is found in case both bins are recurrently inserted. And these recurrent inserted bins are most likely under selective pressure. So this is an indication that also the co-localization is, at least to some extent, under selective pressure. The next question that we asked is, can we also find specific interactions between clusters? And to this end, we clustered the insertion data using kernel smoothing, and we find 777 of these clusters. About 60% of those correspond to previously discovered common insertion sites. This number is not 100% because we used a much lower threshold, because we want to also include clusters of insertions that are not significant by themselves, right? And then we look for interactions between two clusters. We do that actually using the non-normalized data in a 30 KB resolution, and we look at the bins covered by these clusters. So this is cluster A sitting over here, cluster B sitting over there, so we have to look at these values in the matrix. And to take again into account this linear distance bias, we compare those to these diagonals over here. Now we use a Wilcoxon rank sum test for that, and when this is significant, we call this a co-located insertion cluster, or a click. Now, if we do this for all the clusters, we actually identified 874 of these co-located insertion clusters, covering about 480 unique loci, and these loci we call click loci. 309 of them actually are covered by previously discovered common insertion sites, which is an indication that also these known common insertion sites actually engage in these 3D interactions. At the same time, we have 170 of those that do not overlap with any previously known common insertion site. So it's very interesting to consider these and look in their vicinity to see if there's any candidate cancer genes in the region itself, or whether they interact with a region that contains a gene that is interesting as a candidate cancer gene. This is one of the specific examples that came out of our study. This is the Notch1 locus, contains Notch1 gene, and Notch signaling is very important in these hematopoietic neoplasms. And it forms clicks with these 5 other loci, and it might of course definitely be the case that the insertions that occur within these loci actually target genes in the direct vicinity. However, another explanation is that insertions in these loci actually, through long-range interactions, target Notch. And given that Notch is such an important target in these types of tumors, that seems to make sense. Another piece of evidence for this is actually given by the distribution of insertions across these 6 loci. So that's shown over here. So here we have the tumors, here we have the 6 loci, and we can observe a very strong pattern of mutual exclusiveness, which is a typical indication that these insertions actually target the same gene. This pattern of mutual exclusiveness is also observed for many of the other clicks. This is again the Notch1, but also for Icarus and a bunch of other of these clicks that we discovered. And if we characterized this at a more global scale, comparing the significant interactions with the non-significant interactions, we actually see a much stronger pattern of mutual exclusiveness compared to the non-significant interactions, which is a very significant p-value. We also overlapped our clicks with known cancer genes, which we obtained from the cancer gene census. And this is the Venn diagram that we get. I'll walk you through it. First of all, 25% of the click loci overlap with known cancer genes, and this is already much more than expected by chance. But this still means that about 75% of the genes do not cover a known cancer gene. However, of those 75%, more than 50% of them are actually linked to a cancer gene through long-range interactions. And this is much more than expected by chance as well. And so taken together, this suggests that actually the majority of these click loci either contain a cancer gene themselves or are linked to a cancer gene through long-range interactions. So to summarize this, we find a significant co-localization of these cancer-causing insertions, which is more pronounced for recurrent insertions, which is an indication that this is to some extent due to selective pressure. We discover specific co-located insertion clusters, which we termed clicks, and this shows that the 3D organization is important for linking genes to their targets. And so taken together, this really sheds new light on the target genes that actually come out of these insertional mutagenesis screens. All right, so with that I would like to acknowledge a lot of my friends and colleagues that helped me with this work, but I would like to actually acknowledge one person in particular, Sepide Babai, who has been my PhD student until recently. She is about to graduate actually, but she is the person that did all the work for this paper. So with that, thank you very much.
Thank you very much, especially for bringing some 3D structure and otherwise a genomic session, which makes me very happy.
Excellent.
We have questions. Oh, a 3D person.
Yeah, it actually makes me also happy. So thank you for your talk, very nice. I also deal with this type of data, and one of the problems that we all face as a community is how to normalize it properly. There are several ways of normalizing the data, and each of them have a different impact on how at the end you deal with it. So I wonder if your findings are also consistent with normalizing with Amos-Tanai's approach or Laminar
yeah,
approach.
so I didn't mention that, but those normalizations for restriction side bias and stuff, that we already did those before we did the rank-based normalization. So they are actually included in the data. Yeah, yeah, absolutely. Yeah, without those, I think you could not find these results. Yeah, yeah.
Thank you.
Yeah.
Thanks, very nice talk actually. Also quite happy about the 3D genome architecture. My question is, there are more accessible and less accessible regions of the genome. And this might heavily impact both the insertion, but also probably HiC. So how do you cope with that?
No, that's an excellent question. So there is definitely correlation also between the distribution of our insertions and these two compartments, the open and closed. And so we consider that also. But there is definitely also insertions in the closed compartment. So that's why we didn't want to throw them out. But we did do an analysis where we specifically looked for only interactions between open and closed to take this into account. And the results show that even though the significance of these co-localizations is a little bit reduced, it's still very significant. So.
Great talk, thank you. Just a brief question. How do you deal with repetitive elements in the mapping of integration sites and with respect to the HiC?
Yeah, so for the repetitive elements, we don't map integration sites to those locations. We cannot. And I guess the HiC data suffers from the same problem. So those will just be black spots.
Two questions. First, what fraction of the phenomena of cluster will remain if you just look at the data linearly? And second question, is your locations in any way related to the TADs, to the topologically associated domains?
Yeah, so we looked a little bit at TADs, but we couldn't find a very striking correlation in that sense. So it seems to be very different. Your first question on what of the clustering remains. So there is already quite a bit of clustering in the one-dimensional genome because these things are under selective pressure, and that's what we want to observe actually. So yeah, so we didn't really check to see if the total clustering in 3D is random. We just looked at if there's significant co-localization of these one-dimensional clusters. But I guess what you point out is a good thing to reiterate and check for that computationally. Yeah, thanks.
Thank you for the interesting talk. My question is, in your analysis, did you consider both intra and interchromosomal interactions, or only consider one of them? Yeah. So we only considered intrachromosomal interactions, and the main reason is that we really didn't know how to deal with the interchromosomal interactions compared to the intra ones, right? We have this normalization where we can compare it relative to the superdiagonals, which represent a certain distance, but if you look at between chromosomes, yeah, that's going to be difficult. So, yeah, when something is difficult, sometimes it's good not to touch it too much. Yeah, because probably HiC data is generated for a population of cells.
Yes.
The interchromosomal interaction is very dynamic.
Yeah.
But the intra may be more conserved.
Yes, exactly. Yeah, that's another good reason not to touch it too much.
Thank you.
Yeah.
Okay.
So,
one last.
You mentioned that from the cancer genome, what's the difference between cancer and normal for the 3D structure? Is there any?
Yeah, so we use these data as a proxy of the situation before the integration occurred, right? So that's important. Yeah? I guess that after integration, yeah, the genome might change, also the 3D conformation might change. There's actually a few studies about that, but I don't think there's any clear hard evidence and how, it's not really known how this actually happened.
Okay, seems like everybody's happy, and let's thank all the speakers of this very interesting session. Thank you very much.